then with our next presenter. So this is Dr. Heather Knowles. She's an assistant professor and APP associate director of the Cell Therapies division for the Division of Hematology and Blood Disorders and Cell Therapy Center here at the Anschutz Medical Campus. Some background on Heather. After obtaining a bachelor's degree, she started working in a neuroimmunology lab studying inflammation at Albert Einstein College of Medicine. She obtained her PhD in microbiology and immunology at the University of Colorado. While working as a postdoc fellow at the National Jewish Medical Center, she studied mechanisms important for immune system activation in response to infection. She then decided to leave the bench and return to New York City to train as a physician assistant at Weill Cornell Medical and Graduate School. After graduation, Heather started her PA career with our program and has been working at the Blood Disorders Center for us for nearly 9 years. She has been instrumental in developing our CAR T-cell program and how we manage their unique toxicities. So Heather's talk today is immunotherapy, retraining the immune system to seek and destroy cancer. Welcome, Heather.
Thanks, Glenn. So awkward walking around with a walker and becoming a patient, but also enlightening. All right, well, thanks for joining me. I'm the one that always goes over time, so thank you, Meredith, for giving me a couple of extra minutes. Today we're going to do, I like to say, like Meredith had mentioned, a Cliff Notes version of all of immunology, oncology, and immunotherapy in 45 minutes. So I talk fast, so hold on to your seats. I want to do a broad concepts of immunity, which means kind of going back to a few basics. I like to think of my understanding in the clinic, really, the foundation of it is always the mechanisms of disease, the mechanism of a pathology. So we're going to spend a little time there. Then we'll go through the 5 different types of immunotherapy. And then we'll spend some time on immune effector cell therapies and the different types that are in the pipeline, as well as the unique toxicities of all of immunotherapies. So lots to cover. You know, and when I thought about this new Fight Club, I really wanted to get like, you know, a Brad Pitt picture and kind of make it into NK cells versus T cells, but I didn't have the creative juices for that. So you just get the font. We're going to learn to walk before we run. So the reason why I love immunology is the fact that when it's out of balance, you have pathology. I don't love pathology, but I'm very interested in it. And that's kind of the drive of my career over time. So when your immune system overreacts, you have things like autoimmune diseases, which was something I was very interested in my research days, or allergy. When the immune system underreacts, you have things though like cancer and infection. And so it's not a surprise that I ended up in a blood cancer unit where we're looking at cancers of the immune system. So how do cancers hide from the immune system? There's the immunoediting hypothesis about how cancers evolve over time and then kind of break through the homeostasis and immune surveillance that the immune system provides. But it really does try to target it from multiple directions, and that's why immunotherapy is used to overcome those. So, you know, ineffective antigen presentation. When you have dendritic cells presenting antigens, self-antigens, altered, you know, neo-tumor antigens, in the form of antigen presenting cells, they can downregulate, you know, our MHC expression to try to hide. T cell dysregulation. So T cells have checkpoints to kind of control the immune response. Tumors love to overexpress those ligands so that they can turn off the T cells when they come into the tumor or lymph node. Suppressing cells and cytokines. You know, the immune system needs a lot of different positive messages to keep it going. When it gets those negative emails, those negative kind of messages, it turns them off. Defects in innate immunity. So our immune system has a couple of different branches. They all work together collectively. If you can drop off that danger signal and decrease the innate inflammation, you can have an impact on the adaptive immune response. And then there are other mechanisms like tumor angiogenesis and genetic and epigenetic changes that overcome some of the fancier immune systems and ultimately altered metabolism by the tumor. Those are a little bit more relevant in the solid cancer field. So when we think about antigen presentation, it's essential for T cell responses. Dendritic cells are professional antigen presenting cells. There's a lot of different diverse subtypes. They infiltrate tissues, and then they link that inactive and innate or the innate and adaptive response. They prime T cells and influence the behavior of those T cells, so they have a lasting impact on the response. They are essential in generating that anti-tumor CTL response. The T cells, on the other hand, receiving the signals require two signals. They need that first antigen recognition signal, which is a histocompatibility complex or HLA restricted response. So I kind of think of that as like the taco shell. And I don't know, does this point? Yeah. Up here, you've got your little MHC molecules like the taco shell. Then you have an intracellular peptide that's presented as like kind of the stuff in between. And each of these unique interactions with T cell receptors linked to the CD3 zeta chain, which provides a little lightning bolt signal to the nucleus, is that first recognition like this is something bad. Co-stimulatory signals are the signal 2. This is stuff that's activated by inflammation. You get upregulation of co-stimulatory molecules that verifies that need for activation. You need two signals for T cells to actually want to do their jobs. Kind of like how we need 2 RNs to hang a bag of blood. You need to make sure that this is the right person, right patient, right bag of blood. T cells also want that kind of verification. But then to, actually do anything about it, when you start from 10 to the 11th and more possibilities of T cells in your body, you need more than one cell to fight a cancer or fight a virus. You need to grow it into a clone. And for that, you need that third signal or those soluble factors that provide growth and proliferation signals. And those are ones that can also help define the effector and memory functions. And so, for instance, like when a virus is exploding, your T cell response is going from one cell to a clone of cells. And at some point, we could here at the top, we see that viral antigen is going down. The same might be held true in the case of cancer. What you get at the end of this, though, is a series of memory cells that last for your lifetime. And so T cell biology is important for understanding a lot of what we do in immuno-oncology. But those cytotoxic T lymphocytes or CTLs, those are the ones that kill cancer. They're the CD8 effector. They see class 1 molecules, which are intracellular pathogen, peptides via virus or neo antigens. And the proliferation and then recognition of the tumor leads to things like granzyme B, interferon, and TNF alpha, and other types of death ligand signaling. But if we were to allow, if you think back to that graph when the T cells are peaked at this huge response, if we were to allow all of our T cells to do that every single time it encounters an antigen, our body would just be a flaming set of inflammation. So you need a checkpoint signal to kind of control that response. And that comes in the form of what we call checkpoints. So they inhibit proliferation and effector function, and it helps to kind of avoid indiscriminate immunity against self-antigens. So here's our signal 1, here's our signal 2, but then we need to throw on a signal 3, which is one of these types of molecules. So it can decrease co-stimulation and it can help induce death of the cell over time. Now, when we think about B cells have receptors just like T cells. In fact, the B cell receptor is the antibody stuck to the membrane. Antibodies, just for our basics, are 2 heavy chains and 2 light chains. Everything's linked together by sulfide bridges. We've got variable regions, which can be hypermutated to recognize our antigen. But then we have these conserved areas that allow for the effector functions of the innate immune system and complement systems, for instance. And then our antibodies can come in a couple of different flavors. We've got our pentamer IgM, our dimer IgA, our monomers IgG, which is one of our main antibodies, and then IgE reflective in the allergy system. B cells also require 2 signals. So if it's a T-independent antigen, it gets cross-linked by having large polysaccharide kind of chains and creates your signal versus a T-dependent antigen. You've got your B cell stimulation followed by co-stimulation from a T cell, creating the germinal center reaction, somatic hypermutation, and then long-lived antibody responses. So when we think about how antibodies respond, we get that short-lived, low-affinity IgM. It isotype switches to IgG, where we get a better response. And anytime we have a second antigen exposure, we're going to get a much more robust antibody proliferation or secretion, rather. Now, antibodies have a lot of effector functions. We don't think about this in the world of cancer, but antibodies are there to do a lot of good things for us. They are secreted by plasma cells. They can neutralize the pathogen, create opsonization, which would lead to phagocytosis. They can be bound to complement and do the same thing. They can be picked up by FC receptors on NK cells, and then when you get multiple one of those cross-linking an NK cell, it'll send off its death granules to kill a tumor cell. And then, like in the case of allergy, IgE antibodies can be bound to mast cells, and when they're cross-linked by their antigen, can lead to histamine release. So lots of things can happen in response to antibodies. And then finally, I like to think of the NK cells as the secret police of the immune system. They're like, can I see your license and registration? Their job is to basically patrol the the entire body. And they're looking for that, expression of class 1 molecules. They're looking for people, cells saying, here's an example of all my self-peptides. I have not been infected by a virus. I have not been transformed into a cancer cell. They're a really interesting cell, since they're a lymphocyte, really, but they act in the innate immune system. They don't need that antigen-specific recognition. Instead of picking up the taco shell with a peptide, they're just basically looking for the taco shell alone. They have a series of inhibitory and activating receptors. They also can secrete a lot of inflammatory cytokines, interferon gamma, etc. And then they perform antibody-dependent cell cytotoxicity, as exampled by the CD16 molecule on NK cells binding to the constant region of an antibody, which is then bound to, for instance, a leukemic cell or a lymphoma cell. Oops. And then the final part of the immune system, soluble messages, guide responses. These are like the 1,000 emails that we all get every day. So they flavor the experience that's going on in any given part of the body. And when we think about what's been done in immunotherapy, interferons have played a really big role in autoimmune disorders, but also in high-risk melanoma, hairy cell leukemia. The interleukins, of which there are a variety, some are inflammatory, some are immunosuppressive, but we use IL-2 in the setting of metastatic melanoma, TILs, renal cell carcinoma, etc. IL-6 mediates inflammation. Of course, it becomes a very important cytokine when we're thinking about managing toxicities of CAR-T and BiTEs. And then the TNF-alpha family, highly present in autoimmune diseases as well as graft versus host disease. We have drugs that can target that. Chemokines are an important part of the soluble messages of the immune system. They're going to direct the behavior and migration of cells. Transforming growth factor beta family promotes regulation and tolerance. And then all of the hematopoietins that are going to drive proliferation. We use those in the clinic all the time to maintain our patients' blood cell counts. So when we think about immuno-oncology, we can either target the tumor using old-fashioned chemotherapy, autotransplant, which is just high-dose chemotherapy with a stem cell rescue, or monoclonal antibodies, antibody drug conjugates, things that we can mark the tumor for destruction. Or we can target the host. We can use strategies of vaccination, immunomodulation, or checkpoint inhibitors to try to kill our cancer. Or we can target both the tumor and the host. And this is how allogeneic stem cell transplant, bispecifics, and immune effector cell therapy works. So when we talk about the immunotherapy types, really, what is immunotherapy? It's any substance that's going to stimulate or suppress the immune system in some sort of way to help fight cancer, maybe infection and other diseases. Chemotherapy generally refers to chemicals that interrupt cell division. So you have a bunch of classes that do that. Radiations, damages DNA, which offers new antigen presentation. And then targeted therapies interrupt specific signaling pathways. We have tyrosine kinase inhibitors, Bruton tyrosine inhibitors. We have all kinds of small molecule inhibitors, BCL2, et cetera. But immunotherapy is really the new fight club. So we've got targeted antibodies, immunomodulators, cancer vaccines, oncolytic viruses, and adoptive cell therapies. These are the 5 tools in immunotherapy that we can use to fight cancer. So if we go through each of the different types for a bit, naked antibodies are one of the first. They're going to bind to antigens, mark the cell for destruction as shown here. You have your monoclonal antibody like rituximab binding to CD20. It has a signal that attracts, complement or NK cells or a macrophage to lyse the cell. And so we have great examples of this in a variety of blood cancers. So CD20 positive antibodies like Ritux, obinutuzumab, CD19 like tafastamab, alemtuzumab or Canpath targets CD52. So pick a marker, make an antibody, and you can at least harness part of the innate immune system to kill off your cancer. But then we've got antibody drug conjugates. So we're getting a little bit fancier here. Take that antibody and let's put a payload that is going to destroy the cancer rather than just rely on the immune system to help you do your job. So an antibody drug conjugate has 3 components. You have your antibody, a linker, and a payload. It brings some sort of cytotoxic potential to your specific target. And although you can still have things like antibody drug cell cytotoxicity, this adds just an extra layer of protection if you think about it. The linker can help keep that toxic payload stable. It can have cleavable, non-cleavable activities that have been added to it to help it behave in a way that you'd like. And then the payloads, we can choose a variety of payloads: radiolabels, microtubule inhibitors, DNA damage, topoisomerase inhibitors to create our death in the cancer. So this is a good example of how, here's our drug conjugate binding to its surface receptor, becoming internalized, breaking apart, and creating the damage with the payload that it's bringing, causing the cell to die. So we've got some great examples of our ADCCs in blood cancers and myeloma, Blenrep, lymphoma. We have a whole host of options, leukemia even as well. So then, let's take those antibodies further and let's look at bispecific antibodies. We've learned that activating T cells is an important part of killing cancers. If we could just bring the T cell to the fight rather than maybe adding on a cytotoxic payload or relying only on NK cells. So bispecifics were thought of, and in most of the cases, they're T cell engaging. So they're going to have a recognition domain for CD3, which is that important signaling part of the T cell receptor. And then they choose an antigen of your flavor that's marked on your tumor. They, may or may not have that FC portion of it so that they can also engage accessory cells. But they facilitate cell-to-cell interactions, and they facilitate the formation of an immunological synapse. So thinking back to the story about how T cells need 2 signals. They need that first signal of antigen recognition, that second signal that says it's a danger. In this situation, we're doing that without the MHC molecule, and we're creating that co-stimulatory kind of long duration of signaling by bringing things together and clustering it in a way that creates some of the same behaviors where cells are in very close proximity with each other. And then ultimately what this does is to induce the release of perforin and granzyme to kill the cell. And our bispecific antibodies are really, making a name for themselves in the last 2 years. We've had multiple FDA approvals. So it used to just be the tiny old blinatumomab kind of helping us out with our ALL patients, our MRD positive, preparing them for transplant, or our relapse patients. And then we had mosunetuzumab, teclistamab, and now we've got, I'm having a hard time saying all these, talquetamab, I'm going to say Alreflexio. I know. Epcoritamab, glofitamab. I mean, it's a wonderful option to have these bispecific antibodies because they can do a lot of damage and are off the shelf. So what about the checkpoints, right? We talked about how, so all of these therapies so far are trying to utilize T cell activation, maybe utilize the NK cell portion, but what about, turning off the brakes instead of allowing the T cells to die in that normal kind of response to the end of its proliferation cycle? Let's try and keep those T cells going. So if we provide the signal of CTLA4, we provide a brake. If we provide the PD-1 blockade, we dampen or release the brakes. And so immune checkpoint inhibitors, they're negative regulators. They offer sometimes competition with our co-stimulatory molecules, or they dampen the T cells after they've been activated. There are lots of other ones under investigation. And in terms of what's available in the blood cancer world, we don't really use the CTLA4 pathway, but we do have our PD-1 inhibitors, nivolumab and pembrolizumab. These are used in relapsed Hodgkin's lymphoma in combination with other therapies. So, it's pretty exciting how, when you think back to my life originally in the world of science down here, and then to be a part of the clinical world where we have, over 90 different immunotherapeutics available in our toolbox as a clinician is pretty exciting. Immunomodulators, definitely play an important role in blood cancers. So like I said, cytokines regulate maturation, growth, and responsiveness. We've got our interleukins like IL-2 that can affect T cell and NK cell growth. We also have our interferons that are available. And then an important immunomodulator, the IMiDs in multiple myeloma, like lenalidomide and pomalidomide. And then, of course, in development, agonists to other pathways and adjuncts to our innate immune system. Now, if we shift gears for a second, oncolytic viruses, not something that we technically work with in the blood cancer world, but important to know they're in development in one of the 5 types of immunotherapy. So they target the cancer cells. They're usually, you're going to take a virus that targets the cancer cell. It's going to help promote tumor antigen presentation. It oftentimes can be designed to take in additional danger signals to really kind of ramp up the immune response, and sometimes even additional cytokines. Here's an example of one of the oncolytic viruses that's used in melanoma. And then cancer vaccines, another really important arm of immunotherapy, but again, not one that we're using in the blood cancer world, but important to know about it. Just think about it. We use vaccines against viruses all the time. Why can't we just vaccinate somebody who has a cancer? And it's very complicated in terms of what is altered self-antigen, what's a tolerant environment. But we do have a couple of examples of preventative cancer vaccines out there. Gardasil, for instance, targeting HPV to reduce the incidence of cervical cancer. And then therapeutic cancer vaccines, target the established tumor for destruction. There's one example in the prostate cancer world. And then we have finally our adaptive cell therapies, and they're primarily called immune effector cell therapies. And when we think about the immune system and the CBC with the WBCs, right, there are a million different types of white blood cells, from our innate system with our granulocytes and our monocytes and macrophages, our dendritic cells, mast cells. We've got some funky gamma delta T cells, natural killer cells, our B cells, and our T cells. So when we think about immunotherapy and immune effector cell therapy, it could potentially be any of these cells. The big favorites right now are the T cells, the NK cells. There's, work in dendritic cell and gamma delta cell therapies. But we'll be talking about in a little bit more detail are the genetically modified T cells and the natural killer cells. Importantly, in our world of solid tumor, the tumor infiltrating lymphocytes are gaining some very big, exciting traction with possibly an FDA approval this year. But there's also things like viral specific cytotoxic T cells. We've had a clinical trial looking at that in the post-allo transplant patient population. So what if you could help control CMV reactivation in a patient that has resistance to our antiviral? So these possibilities in terms of using immune effector cell therapy can be broad, ranging outside of even cancer. So I love pictures, and I love thinking about how, if you could only just take a movie inside your blood and see how these cells are working, that'd be pretty cool, but also maybe a little gruesome. So when we think about the immune effector cell research landscape, I like this figure because it really demonstrates the growth of immunotherapy in the course of T cell therapy. 2020 to 2022, just the number of clinical trials and products that are under development. TCR T cells, these are more tumor specific antigens, our NK cell and combination therapies, our TIL therapies, and then some other cool targeted therapies. So really, when we think about that figure with the explosion of antibodies, now we have this explosion in cellular therapies. So what is adoptive and what is immune effector? So adoptive just means you're transferring cells into a patient from somewhere else. And immune effector says that those cells are of the immune system and mean that they're going to do something about it. So for instance, TILs they're tumor infiltrating lymphocytes, meaning we need to remove the tumor from the patient. We need to dissociate the tumor and isolate the T cells. We need to give them some enrichment and re-educate them with some inflammatory cytokines to get them revved up and pissed off, and then put them back into the patient. They'll go and find the patient's tumor, hopefully because it's a polyclonal population looking at tumor-associated antigens. Or we can engineer T cells, and we can take the T cells from the patient's blood. We can, through leukapheresis, and then we can modify the cells. We can genetically engineer them any way we want. Do we want to put in a new receptor? Do we want to knock out an old receptor? Do we want to add some cytokines? The imagination is limitless, and then we need to expand those cells and put them back into the patient. And, right now we have FDA approvals of 6 different CAR T cells. There are TCR T cells under investigation. And then when we think about the peripheral blood, we can take this from the patient, or we can take this from a stranger, a healthy donor. Can we do allogeneic cells? But ultimately, we need to create a clonal population that is specific for their cancer. So when we think about genetically modifying, there's already the existing T cell receptor. It has an alpha unit, a beta unit, has some epsilon others, but the zeta unit is the one that sends the signals, and it hooks up with intracellular proteins, really kind of zap the email, if you will, to the nucleus of the cell to change its behavior. And this is our tumor antigen. So here's a class 1 with a beta-2 microglobulin, and then the little peptide in the taco shell, if you will. Versus CAR T cells are completely different. We're not using any of the MHC. We're using the variable region of an antibody that hooks up to any surface antigen of choice. And then we artificially give it a bunch of inside stuff so that it can have the behavior of this receptor. So when we think about augmenting the TCR for tumor-specific antigens, this is what we call a TCR T cell. They're going to be MHC restricted. Versus chimeric antigen receptors, the chimera is, going to be different, and it's going to be MHC or HLA independent. A really cool technology that I was looking at because of our med-onc colleagues and some of the collaborations, just trying to figure out how can we do these clinical trials on campus, thinking about these T antigen coupler therapies, which again are going to be antigen independent, so kind of like an antibody binding domain, but then maybe it has another domain that kind of binds to the T cell receptor, and then also includes some intracellular signaling. So again, you can imagine whatever you want to do and make it happen. You've just got to do all the work in the lab. So the generations of CAR development, when you think about this, has been going on for a long time before we ever got a chance to put this in humans. But ultimately, they use the technology we learned from making antibodies to bind to our cancer antigens, and then steal the zeta chain off of the T cell receptor. Oh, and then go ahead and steal signal 2 from one of their co-stimulatory domains, and that's what we call a chimeric antigen receptor. Now, over time, we had different generations of this. They first kind of made it with just the CD3 and realized that's not enough. T cells don't respond to one signal alone. And then they did the second generation, which added those co-stimulatory, and that's where all of our FDA-approved CARs currently exist. Third generation, they're looking at adding double co-stimulation. Fourth, you can add in cytokines, sometimes called trucks. And then, 5th generation, where we're looking at allogeneic CAR-Ts, where we're knocking out the endogenous T cell receptor so that we can avoid graft versus host disease. So lots happening in the world of CAR T cell development, but of our FDA approved for non-Hodgkin's lymphoma, we have for our aggressive and follicular, and then for our mantle cell. Our ALL, we've got 2 products depending on your age. I work in adults, so we primarily work with Tecartus. And then multiple myeloma, we've got 2 products, Abecma and Carvykti. So what about gaining better controls of CARs? We're getting a lot of work in the field now looking at because CAR T cells are pretty potent therapy. Can we do something to kind of lessen the toxicity? And so they're looking at ways of tuning the affinity of the CAR so that it can maybe reduce that off-target toxicity. And there's a CAR in clinical development that's showing some great response in humans. We can turn the CAR off using suicide genes or elimination markers. So think about if you have a patient with a severe toxicity and you want to just turn things off, can we throw in an adapter to turn it off? There's also split CAR dimers, TET-on regulation. So can we give somebody a drug to turn it on and off? And then logic gates, can we say that you have to have both or one or the other? So lots of things in development in terms of CARs. But one important one to think about that I think a lot of us are gaining, our institutions are picking up on these allo CARs is this idea that you've had a really sick patient who's gone through a lot of therapy. Their T cells have been beat up by multiple rounds of chemo, immunotherapy, and probably don't behave like a normal T cell. So if we could take some normal T cells from any human volunteer and say, hey, can I draw your blood so I can make some stuff in the lab and cure some patients with cancer? I'd volunteer for that. But, then we have that graft versus host disease problem. When we think about transplant, we've got to match these patients. And so how do we get around all of those? There's lots of work in the technology of that. So to overcome graft versus host disease, we've got to kind of get rid of the TCR by, this, where is it? Here we go. Here's our TCR. We do this, genome editing to get rid of the alpha locus, which will inhibit the cell from being able to express that receptor. We have to think about being rejected by the host immune system. So if our HLA doesn't match that taco shell, if it's not the right, HLA for the host you're putting in, so we got to get rid of that. One way to do that is to get rid of the beta-2 microglobulin molecule so that you're not expressing your HLA. So get rid of that. Maybe we want to avoid NK cell allorejection, so we need to add in some inhibitory HLA-E. And maybe if we're going to use lymphodepleting agents so that we can reduce the amount of fratricide T cells killing T cells, we need to often eliminate CD52. So these are highly genetically modified cells so that we can reduce these hurdles in the patient. Importantly, allo NK cells, they don't carry the same risk of GVHD. So there's a lot of work in what can we do to an NK cell to potentially make an allotherapy. So speaking of NK cells, our NK cells are, like I said, the secret police of the immune system. They have a lot of options that are available to them in terms of how they interact with tumor cells. So you can imagine that we can add inhibitory receptors, add activating receptors. We can alter the CD16, which binds to antibodies. We can make that affinity stronger so it holds on to those antibodies better and creates a tighter interaction with the cancer cell. And overall, the benefits of NK cell therapy is better safety. They don't secrete the same cytokines that T cells do, so you won't have cytokine release syndrome, potentially neurotox, and the graft versus host disease. So a good option for off-the-shelf manufacturing. Unfortunately, maybe sometimes not the greatest in terms of efficacy. So lots of work being done in the NK cell field. When you look at just the number of NK trials with antibody targets in combination therapy, just a growing field with lots of options and options in solid cancer as well. Now, our final type of immunotherapy is dendritic cells. Again, not one that we use in the blood cancer world, but in our solid tumors, it's nice to be able to take out some and dissociate our dendritic cells, add new antigen, maybe add some adjuvant, create, sorry, I'm going in the wrong direction, create a more activated dendritic cell, put it back in the patient with the hope that it will educate T cells. Or you can have, vaccines that we're adding certain adjuvants, antibody conjugates to try to teach the dendritic cells inside the patient how to do a better job of priming T cells. So overall, I would say in terms of immune effector cell therapy and in the world of blood cancer, CAR T cells are winning. And as we look at the number of clinical trials worldwide, over 900 different clinical trials, it's a very active study of investigation, both from NK cells, T cells. We're doing it in blood disorders, solid tumors, lots of toxicity management clinical trials, people looking at auto versus allo, looking at peripheral blood, cord blood. Can we create T cells from that? Are we doing an IV infusion? Are we doing intratumor delivery? So like glioblastoma, can we put a CAR T cell in the brain intraoperatively and help get rid of residual disease after a dissection? So the final part of the talk is the toxicities part. And again, we just talked about like every cell of the immune system and all kinds of ways of interacting with it. So you can imagine there's going to be a lot of toxicities to talk about, but I'm just going to hit the highlights. But I think important for clinicians is to think about, it's a new drug that I'm not familiar with. So let me ask myself the question, what is the therapy like? What is the target? How is it delivered? What are the off-target interactions? What kind of a dose or genetic engineering has been done? And do I understand what kind of activation of bystander cells might be taking place? Then when I think about my patient, I'm like, what is their tumor burden? What's the location? Do they have a big mass around their trachea or do they have a big mass in their leg? It could maybe make a difference in terms of what type of clinical toxicity you see. What's their performance status? Is it a young 39-year-old or is it a frail 78-year-old? What are their pre-existing conditions, baseline inflammation, and their baseline coagulopathy status? So, toxicities can be immediate or delayed. When we think about immediate or early toxicities, I think primarily of infusion reactions, but we also have things like hypersensitivity, capillary leak, cytokine release, neurotox, HLH, tumor lysis syndrome, DIC, tumor flare, hepatotoxicity, rashes, infections. A very wide range of things we're not going to talk about in any specificity. But then there's delayed and prolonged toxicities that affect our patients. The myelosuppression is key. It impacts the infection risk. A lot of our antibody drug conjugates can bring on, have the same types of neuropathy that, general chemotherapy can offer. So we need to screen for that. The autoimmune reactions, secondary cancers, and of course the financial impact for the patient. So in terms of infusion and hypersensitivity reactions to antibodies, I thought it was important to just name that we've got basic cytokine release type of reactions, and that's that dose or infusion rate dependent reaction you have with the patient. We can definitely re-challenge those. When we think about type 1 sensitivity or the classic allergic reaction involving IgE, this is anaphylaxis. This is not one that we re-challenge. And then there are some delayed hypersensitivity reactions like serum sickness, for which we can manage with steroids. Our antibody drug conjugates, like I said, it varies depending on the payload and then, of course, the target. So I always say, always check your package insert because, things like ocular toxicity, very unique to Blenrep, rashes, pulmonary toxicity. So, check your PI. Checkpoint inhibitors, automatic, when you think about, you've released the brakes of the immune system. Remember the first slide, I talked about the balance of the immune system. When it overreacts, what does it cause? It causes autoimmune diseases. So most of our checkpoint inhibitor toxicities are related to this autoinflammatory toxicity. They're transient, they can be resolved when you stop the checkpoint inhibitor, but also think about the half-life of an antibody in the patient is going to be weeks to months. And then when you're combining checkpoint inhibitors, you're increasing those risks. So when you think about what you're doing with, keeping kind of the growth of your tumor-specific cells by blocking their checkpoint, you're creating these big polyclonal responses. But there are autoreactive clones in our body, and those are the ones that are also increasing. It creates the autoimmunity. So when we think about some of the incidences across the PI from Pembro to Nivo, dermatologic toxicity can be high, GI toxicity is high, and oftentimes these can be after having several doses. Cytokine release syndrome, our big favorite toxicity. It has been described for 30 years, but really has become the forefront of excitement now, particularly in the setting of COVID. I always say, like, before COVID, I'd call the ICU and say, I have a CAR T patient that has cytokine release syndrome, and we're given tocilizumab, we might need a transfer. And they're like, what? What is it? And now when you say cytokine release syndrome, they're like, do they have COVID? No, they don't have COVID. They just got a CAR T cell. What's a CAR T cell? Okay, let's take a break and we'll talk about it later. But it can be triggered by antibodies, like I mentioned before. How many rituximab reactions have you handled in your life? These are rate dependent and easily managed with steroids. Our T cell engaging therapies are the primary ones that we're looking at now with bispecific antibodies and CAR T. It also happens in haploidentical stem cell transplant. In the absence of immunosuppression, those first 5 days, you classically can get a fever at around day 2 or 3, right before you get your post-transplant cyclophosphamide. And then, like I said, infections. So what is CRS? It's going to be a big fever, constitutional symptoms, and then depending on the severity, you can have depressed cardiac function, hypoxia, and end organ toxicities. In terms of onset of CRS, this is for CAR T cell slide, when it has my kind of median days for the different drugs. But we also need to think about it in the setting of bispecific antibodies. Well, what is immune effector cell neurotoxicity, or ICANS? It's just, manifests as an encephalopathy. We have seen fatal neurotoxicity in CAR T trials. Most of our patients have some sort of cognitive impairment or altered level of consciousness. And in a few patients, we've had just purine aphasia, which is a highly specific symptom. ICANS in general can happen concurrently with CRS, but most of the time it's happening after CRS has occurred and resolved. And then in some patients, we've seen delayed. Depends on the product in terms of the median onset, in terms of, like, we have, for instance, Cilta-cel, where we have an onset at day 8 versus ide-cel with a day 2. So it depends on your product. It depends on the target. Always read your package insert, look at your trial data. But in general, CAR T cells have the class effect of CRS and neurotox. And this is my general summary slide of our lymphoma products, our myeloma products. We've got our CRS all grades and grade 3 or higher. Neurotox all grades are grade 3 or higher. And when we think about in products that can be given for both non-Hodgkin's or mantle cell, the incidences are slightly different. The same is true for our ALL patients versus our non-Hodgkin's lymphoma. ALL reacts more frequently than the lymphoma patients. But importantly, bispecifics share cytokine release syndrome, just like CAR T cells, but at a different level. It's a much more reduced, but still an important thing. And cytokine release syndrome is fairly common across the board, depending on what disease you're treating. The high grade events, as you can see, is much lower than when we think back to the CAR T cells. ICANS is a fairly rare event. They talk about neurologic events, headaches and things like that are going to be higher. But then there are some unique and other toxicities that are worth looking at, and again, look at your package insert if you're unfamiliar with the drug. Now, following CAR T cell therapy, we have a lot of other toxicities that are things we have to worry about. The same is true in bispecifics, hypogammaglobulinemia, that impaired B cell. When we're targeting CD20, CD19, we're oftentimes getting rid of healthy B cells. Patients can have prolonged cytopenias. We're starting to pick up on these late neurologic and psychiatric events. And then obviously screening for subsequent malignancies, since most of them have received chemotherapy, including the lymphodepleting chemotherapy. So in terms of our hematologic toxicities, again, therapy-related factors, patient-related factors lead to things like neutropenia, cellular immune deficits, humoral immune deficits, and the types of infections that can be associated with these. And then I'm going to end on this, which is, when you think about all the surface markers and then their different behaviors, whether you're an AML cell, lymphoma or myeloma, when we've got macrophages, NK and T cells, can consider the possibilities. Like I said, the imagination is where our limitation is. So in conclusion, there are many ways to alter the immune system to recognize and kill cancer, which is exciting, but also confusing. Immunotherapy for blood cancers is rapidly growing. Will it replace traditional chemotherapy eventually? I believe, yes, it will. I think we won't be giving traditional chemotherapy in 10 years. We do have immune effector cell-based therapies. They are a living drug. A single infusion is our hope. And we have 6 FDA-approved products for that. Bispecifics are gaining ground as off-the-shelf products. And now the fight is do we do CAR T or do we do a BiTE? There's still a lot of research to look at and patient outcomes to evaluate before we make that decision. And then finally, I like to say, like I said at the beginning, understanding the mechanism of your drug really helps to provide the clues that you need about their toxicities and their efficacy. But importantly, CRS is our common one associated with both our CAR Ts and our T-cell engaging BiTEs. So with that, thanks for inviting me and thanks for letting me talk today. Happy to take questions or comments. Lucy.
How do they make the products in the lab? Like, how do they do
Which products are we talking about?
anything? Like, is it a Lego? Like, how do they?
Making monoclonal antibodies is done in a variety of ways. Usually you have hybridomas or cell lines that can secrete all of this, and then it's purified. You can do it both from bacteria, from cells. And so it's just a matter of, again, the cool thing about harnessing biology to make biology. So our bodies, cell types are meant to secrete antibodies. So you can make hybridomas, which are plasma cells that just pump out lots of antibodies. And we have different cell types from, chicken, hamster. I think about all the different ones like in the lab in terms of how they manufacture, like rituximab. I can't tell you if it's bacteria-based or a cell-based system, but ultimately we're just, again, harnessing your own biology. In terms of CAR T cells, you're taking your patient's T cells, but what you're making in the lab is the virus. The virus has all of the genetic, it's taking a plasmid with a bunch of DNA that you've designed and created, incorporated into the virus. You have to create lots of virus. There are, lots that are made. They're checked for their efficacy in terms of infectability. So, all of this is done in big bioreactors and yeah. Meredith.
Awesome talk, first of all. Thank you for making it feel like, oh, I kind of understand this. It's not as overwhelming. So one thing that I've really struggled with has been taking care of patients. So in thinking about CARS, I feel like, okay, cool. CRS, neurotox, got it. But these BiTEs that we infuse over, several different instances. One of the things I've wondered about is how does your risk of CRS change over time with each infusion? I haven't quite, I feel like, mastered that. And I feel like I've seen a few different approaches to managing that.
Yeah.
And so I was curious if you had some thoughts of wisdom on that.
When you look at how the bispecifics are delivered versus the CAR T cells, they're done in a dose escalation manner. So I think, we'd like to just give one dose. The problem is would the patient tolerate that dose? So they're given in a dose escalation manner. When you look at the cytokine release syndrome incidence, it's going to be highest on the first dose, even though it's a small dose. And the incidence decreases during the step-up dosing profile. And then by the time you get to that target dose, you've managed most of the cytokine release. Now, what's happening in those situations? I can only hypothesize that you're actually killing off some of the tumor and you're reducing the amount of antigen recognition so that you're creating, like when you think about bulk, the bulk is reduced, just like when we pre-phase a patient before starting a chemotherapy with, for instance, steroids. And so by doing a small dose, reducing some of the bulk, we're reducing the incidence of CRS over time. The amount of recurrent CRS that's that's the one to look at. Like, okay, my patient's coming in for cycle 2. You're scanning the package insert. Look for what's the recurrent CRS rate. Most of the time, they're going to be single digits and even lower. But I think that this is the potency and the volume of antibody that you're infusing into the patient and the bulk of their disease is going to make a big difference. I think over time, getting patients treated earlier before the bulk of their disease is larger is going to be more successful, which has been the case as we look at the success in ALL with CAR Ts and even lymphomas. If we can bring the bulk down, the toxicity is less, with the efficacy is better. Carrie.
Do you think that bispecifics are going to replace CAR T?
I don't think they'll replace it, but I think it's going to take a while for them to replace CAR T, and partly because there are patients who want to come in once, maybe go through 30 days of monitoring, and then be done with their therapy. They don't want to come in every 2 weeks. They don't want to come in every 4 weeks. They don't want to say, oh, I'm tied to an infusion center for the next, 10 to 20 months or even longer. So until we have the ability, to make bites a shorter duration of therapy, I don't think that they'll take out CAR T completely. I think there's a role for having that single drug living infusion. The thing that I always think about, I would really like to figure out a way to track the CAR Ts in the patient. I know that we have looked at, and there is data to suggest that they don't persist. But that's only to the level of detection that we have, right? So are there any persisting CAR Ts, memory CAR T cells that are living in a lymph node that you can't pick up on a flow-based assay? So I still have this belief that they are a persisting single drug, and it's a matter of how can we reawaken that if it's there. Lots of things to investigate, though.
Hey, Heather, I also concur. That was a great talk. So these allogeneic off-the-shelf CARs, it looks like there's a lot of ways a lot of thoughts on how we can mitigate graft versus host disease. I guess for one, do you think that's possible to truly mitigate graft versus host disease with these, off-the-shelf CARs? But then also, is it possible kind of through the similar mechanisms to reduce toxicity, like the neurotoxicity and CRS and other things we see with CAR T cell currently?
So the toxicities associated with CAR T cell, I think, really are inherent in what is the construct and the chimeric and a receptor in itself. So I think that, it's not just the CD28 domain that creates a higher level of potentially toxicity, but really the linker and the transmembrane domain. And it really gets to this idea of how much immunological synapse is engaged and created through that construct. And so there's, Terry Fry, who's worked on CD20 and has CD22 and has made in the lab several different types where they've tried to shorten or lengthen linker and have seen vastly different toxicities, also vastly different efficacies. It is really a fine line in terms of how much antigen engagement and how much synapse have you created that creates the strength of signaling enough to change the behavior of the cell. In respect to the AlloCARs, I've read through many of the protocols. We have a couple in the pipeline for an allo for myeloma and an allo for lymphoma that are coming up probably next year. And, it just blows my mind the amount of genetic manipulation that when I think back to my days in the lab making retrotransgenic animals and, making viral constructs, the idea that you have to knock out the T cell receptor, introduce an inhibitory receptor or introduce or knock out the CD52 receptor and express a CAR without altering the, intrinsic DNA of the cell so that it doesn't become cancerous in itself. I mean, it's a lot of manipulation, and I'm not quite there in believing that allo-CAR Ts are going to be an easy off-the-shelf option, but the idea of having a healthy T cell as your baseline, certainly institutions like ours early on, where we had a period of time where we would bank some of the cells from our patients. We would go ahead and collect them, bank them early, knowing that they had high risk of relapse, and then going back and using the Novartis product for CAR T development. That might be the way to go, but really, do we have time and energy and money to bank every cancer cell patients' cells at the time of diagnosis. I don't think that'll ever be possible either. And that's where I think bispecifics are going to be more engaging in the field, because it's the off-the-shelf product without the GVHD risk. That's my opinion. Anything else? All right. Well, thanks for your time.